Hello everyone. In today's video, we will be talking about genetic mutations. Yeah, like the things you heard about in Teenage Mutant Ninja Turtles or any of the sci-fi movies that you might have seen. But instead of talking about those fictional representations of mutations, we're going to be talking about how in real organisms, changes to the DNA of the organism can affect the organism. So, what is a genetic mutation? Again, we hear it all the time. You probably think of a mutant creature that's completely weird and messed up, but genetic mutations are actually incredibly common and happen all the time. So, a mutation is any mistake or change in an organism's DNA. That means that any slight change or variation in an organism's DNA is actually a mutation. It doesn't necessarily mean that you turn into this big, weird monster like we see in the movies. So we know that organisms' genes, which are the small sections of our DNA, are made up of these long sequences or codes. What happens in a mutation is that something causes a change to any given part of this code. So for example, we can see here that along this stretch of whatever organism's DNA that we're looking at, it had a code of CTAG. For some reason, which we'll talk about in a moment, a mutation took place, changing that sequence to CATG. So we can see that there has been a change or a shift in the code. That A and that T were switched around, causing the code to be different. That is what we call a genetic mutation. Now, what causes these mutations to happen? Now, I'll keep going back to the movies because that's where we hear this word. You've probably heard of, Spider-Man being bit by a radioactive spider and that causes a mutation, or being dropped into a vat of some chemical causing a mutation. That's really not how mutations happen in general. Most, meaning almost all, mutations actually are just random mistakes that happen when DNA is being copied in cells. So think about when you're writing a sentence or writing a whole paragraph, you might accidentally spell a word wrong, or you might write a D instead of a B because you're writing quickly. Cells, as they are replicating or copying DNA, make mistakes all the time. This is where genetic mutations arise or where they come from. Now, like we saw in the movies, there are some rare cases where environmental factors, meaning things on the outside, such as radiation or harmful chemicals, can cause mutations. This, however, is actually quite rare and is not the cause of the vast majority of genetic mutations that we see. So we remember, moving forward, before we talk about types of mutations, that a mutation is any change or mistake in an organism's genetic code. We see another example here where this original sequence along the gene was TGGCAG, and a mutation has taken place, changing it to TGGTAG, seeing that C has been changed or shifted to a T. Even that small, tiny mistake, just one letter, is a mutation that can potentially have serious outcomes for the organism. That's what we'll go ahead and talk about now. So we know genes are those sections along DNA that give the instructions or the directions for which proteins the cell should make. Some mutations actually change those directions or edit that recipe enough to cause a different protein to be created. We also remember from our previous video that proteins and the functions that they perform determine the trait that the organism will exhibit. Some mutations or some changes in that code on the gene actually cause different proteins to be produced that can eventually cause a change in the trait or the observable characteristics that we see in the organism. Some of these traits or these mutations that cause different traits are beneficial, meaning they're good for the organism. Other mutations lead to effects that are harmful or bad for the organism. And then even other mutations can cause changes that are neutral, meaning they have no effect on the organism. So now we'll jump in and talk about these three different types of mutations. Again, they're beneficial, harmful, or neutral. So a harmful mutation, you probably can start to piece together from the word harmful, is a mutation that has a negative effect or effect that's bad for the organism. Harmful mutations are mutations that change the protein, resulting in a new protein being created and hence a new trait that is harmful to the organism or bad for the organism. So this looks like a mutation on the gene, so a change in that genetic sequence, that code, that causes a different protein to be produced, and the function of that protein causes a new trait to be observed in the organism, which is actually harmful or bad for the organism. Let's look at an example. So there's a species of desert rabbit, you can see here in the picture, that have a fur color gene. Most rabbits on their fur color gene has a set of directions or a code that tells their cells to produce a protein called brownocells. This isn't the real protein name, I'm just simplifying it for us. This protein causes the cells to create brown fur. This is great for the organism because these rabbits are then able to hide from their predators, which allows them to survive and reproduce, making more bunnies and being very successful evolutionarily. Now, some desert rabbits of this species have a mutation on their fur color gene. This change in the code causes the rabbit cell to produce a protein called white-globin, which then causes the trait of fur color in this rabbit to be white fur. You can probably start to see that that would be very bad for this organism because this rabbit with the white fur is no longer able to camouflage well in its environment and escape from predators. This means that they will get eaten more frequently. They won't be able to survive and reproduce. So this overall is a very harmful trait to this organism. The next type of mutations we'll discuss are beneficial mutations. You probably can already guess that beneficial mutations are mutations that have a positive or good for effect on the organism. Beneficial mutations are mutations that cause a change in the protein, which results in a new trait that is beneficial or good for the organism. So this means that a mutation happens on the gene, a change in that code or that sequence, which causes a different protein to be produced. That protein's function leads to a new or different trait, which is actually beneficial or good for the organism. Now let's look at an example. So in humans, we have a gene called the ACTN3 gene, okay? On this gene, this causes our cells to produce a protein called alpha-actinin 2, which is a protein related to muscle function, okay? Some people, not the majority, but some, have a mutated version of this gene, which gives directions to produce a different protein called alpha-actinin 3. Alpha-actinin 3 actually is a protein that allows for even better muscle function, meaning that people that have this protein created in their cells are stronger, faster, they're better at lifting weights, running, all of these things. This is a beneficial mutation to humans because this allows these people to be faster, stronger, more successful in athletics, and in an evolutionary standpoint, better to survive. If we were still cavemen, stronger, faster cavemen survive because they can hunt better, they can escape from predators that they actually have to live amongst, okay? Meaning that this is a beneficial or good for the organism trait, giving us an example of a beneficial mutation or a mutation that causes a protein to be produced that actually leads to a trait that is better for the organism than the non-mutated trait. Finally, we'll talk about neutral mutations. If you know what the word neutral means, you probably can already guess what this type of mutation is. So a neutral mutation is a mutation that has no effect. It's neither good for nor bad for. It is neutral, okay? It does not affect the organism at all. Now, there are 3 different ways that a neutral mutation can come about, or 3 different types of neutral mutation. The first time that a neutral mutation takes place is when a mutation takes place but it does not even cause a change in the protein. So the change in the sequence is not enough to change the recipe, so to speak, enough to cause a new protein to be produced. Obviously, if it's the same protein, there will be no change in trait, hence there's no effect to the organism. Second instance that can be considered a neutral mutation is when a new protein is produced, but the function of the protein doesn't change the trait that is exhibited. So a new protein is produced, but ultimately that new protein does not affect any trait in the organism, hence no effect on the organism from this mutation. Finally, there can be a production of a protein or a different protein that does lead to a new trait. However, that trait does not either positively or negatively impact the organism. Hence, the mutation as a whole is neutral. So again, we have three instances of neutral mutations. We have a mutation that takes place, which does not cause a change in the protein produced, meaning the same protein is produced, and hence there's no change in the trait, no effect to the organism. Our second instance is when that mutation takes place, causing a different or new protein to be produced. However, the function of that protein is not different enough to cause any change in the trait. Hence, again, no impact on the organism. The third instance is where there is a mutation that causes a different protein to be produced, which does lead to a new trait, but that trait is not positive or negative, not beneficial or harmful to the organism, meaning overall it has no impact on the organism. Let's look at a couple of examples. First example of a neutral mutation is one where, again, a sequence on that gene is changed, a mutation takes place, but it does not change the protein that is produced. So we can see in our example here that we have initially a TTC-AAG sequence, okay, which causes a protein called lysine to be produced. We can see that in this mutation, it's mutated to TTT-AAA. We can see that there has been a change in that sequence. However, the recipe or the directions still lead to lysine being produced. No change in the protein, hence no change in the trait, no effect on the organism. Another example here is, you can pause the video and read through this if you'd like to, but humans also have a gene called, for short, the PTC gene. This causes the production of a protein that allows a person to actually taste bitter flavors. So Sour Patch Kids or rotten milk, people with this protein actually are able to taste that flavor. Some people have a mutated gene on this PTC gene, which causes a production of a different protein, okay, which makes these people unable to taste bitter flavor. So in this case, we have a mutation that causes the production of a different protein and a change in trait, that ability to taste bitter or not to taste bitter. However, being able to taste bitter food or not is not a trait that actually impacts the organism, the human, in a positive or negative way. Hence, even though there is that change in trait, we still would call this a neutral mutation because it doesn't impact the success or the survivability of the human, sort of just, I can taste bitter or I can't. So this is another example of a neutral mutation.